رحاب فؤاد ورابة, lecturer at Oral Medicine, Periodontology, Oral Diagnosis and Dental Radiology. Ecology is a scientific study of the interaction among organisms and their environment, such as the interactions organisms have with each other and with their abiotic environment. What is the meaning of abiotic environment? There are living components of the environment such as air, oil, water, and soil. Ecosystems are composed of dynamically interacting parts, including organisms, the communities they make up, and the non-living components of their environment. Eco-niches, the set of biotic, which is the living components, and abiotic conditions in which a species is able to persist and maintain stable population sizes. Oral microflora refers to the community of the microorganisms coexisting in the oral cavity as its primary habitants, includes bacteria, viruses, protozoa, fungi. The mixtures of organisms regularly found at any anatomical site is referred to as a normal flora or resident flora. Some researchers prefer the term endogenous microbiota. More than 300 morphologically and biochemically distinct bacterial groups or species of bacteria are recognized as normal inhabitants of the human oral cavity. They are classified according to its biomass to high biomass sites, non-shedding surface, which are present on non-shedding surface as supragingival tooth surfaces, subgingival tooth surfaces, shedding surfaces, the tongue. Low biomass reservoir sites is a low number as shedding oral mucosal surface, buccal, palatal, external gingiva floor of the mouth, and here saliva as a transitional zone. For the high biomass level of bacteria, which are found strongly attached to the tooth, supra and subgingival, and the black, which is a low mass, which are present in the external surface of gingiva, and it's not fixed. It's always changing. Okay? Also, the bacteria are classified according to its reaction to the stain, which may be gram-positive or may be gram-negative, according to the ability of the cell membrane to differentiate. Also according to its shape under the microscope, which may be cocci or rods. The growing ability of the microorganism according to its ability to grow in the presence or absence of oxygen. When it's called aerobic, it grows in the presence of oxygen. When it's called anaerobic, it cannot grow in the presence of oxygen. Facultative, it can grow in the presence or absence of oxygen. Cabinophilic, as the growth of the microorganism is stimulated by CO2. Also, it's classified according to its ability to use fermented carbohydrates. To ability to use and to ferment carbohydrates. Saccharolytic, when able to ferment carbohydrates. Saccharolytic species which cannot ferment carbohydrates. The origin of the oral microflora. The microflora present in our mouths is acquired from birth. Newborn babies have no bacteria in their mouths. Over time, the oral cavity becomes colonized by the strains of bacteria that eventually form the resident microflora, which establish a permanent population in the mouth. Over time, as the individual is further exposed to the external sources of bacteria, the biodiversity of the oral cavity increases to the point where stability is reached. This is termed the Climax community. The sources of microorganisms. The sources of these microorganisms mainly comes from the saliva, although it also includes the food and water consumed by the individual. This process takes place within the first few hours of life. Majority of the children obtain their resident microflora from their mothers, as they often possess identical strains of bacteria. This is known as vertical transmission. whereas horizontal transmission also takes place as children interact with their peers and later in life between spouses and partners. The settle of all these econiches occurs relative stability of the system by the microorganism called the Climax community. The stability of the community is based on the hemostasis, including compensatory mechanisms against the effect of various factors. At birth, Streptococcus salivarius is dominant and may take up 98% of the total oral flora until the appearance of the teeth at 6-9 months in human. Periods of formation of oral ecosystem: the neonatal period, mostly aerobes, Candida albicans, obtained from the mother and surrounding area. Periods before eruption: facultative anaerobes and anaerobes, Streptococcus 90%, actinomyces, Candida albicans. Rahaf Fuad Ghuraba, lecturer at Oral Medicine, Periodontology, Oral Diagnosis and Dental Radiology. The eruption of the teeth during the first years led to colonization by the Streptococcus mutans and the Streptococcus sanguis. This bacteria requires a non-desquamating, non-epithelial surface in order to colonize. They will persist as long as teeth remains. Other strains of Streptococci adhere strongly to the gum and cheek but not to the tooth. Periods after eruption, you will have anaerobes, actinomyces, lactobacillus, veillonella, fusiformis, spirochetes, candida albicans. Oral flora changes with ages. Time during the lifetime. At the newborn period, oral cavity sterile, soon colonized by facultative and aerobes organisms, example Streptococcus salivarius. At 6 months periods, flora becomes more complex and include anaerobes organisms such as veillonella species and diffusiform bacteria. At the period of tooth eruption, increase in complexity. There is Streptococcus sanguis, Streptococcus mutus, and actinomyces viscosus appears. New habitants include hard surface and gingival curvicular fluid. Child to adult, various anaerobes frequently found inside the patient mouth, members of the bacterium. The Bacteroideca spirochetes isolated more frequently. Loss of tooth, disappearance of Streptococcus mutans, Streptococcus sanguis, spirochetes, and many anaerobes which is firmly attached to the tooth through dental pellicle formation. Dangerous reappearance of bacteria able to grow on hard surfaces. Variations in the microflora. Everyone has microflora, but it varies between people depending on the condition of their oral cavity. Not all individuals would have the same microflora profile, and also not all bacteria are ubiquitous in all individuals. Ubiquitous meaning a widespread in all individuals. Factors affecting the growth of the microorganism in the oral cavity: temperature, Redox potential, anaerobiosis, pH, nutrient, endogenous and exogenous from the food you are eating, host defense, innate and acquired immunity, host genetics, a change in the immune response, antimicrobial agents and inhibitor. Temperatures constant. In the gingival cervicular fluid is about 35 to 36 Celsius. Periodontal bucket with active disease have higher temperature compared to the healthy, can reach to up 39 degrees. After ecology of the periodontal bucket by alter for bacterial gene expression and the possibility of the competitive of the individual species, downregulate the expression of major proteases as well as gene coding for major subunit protein of pamprea in the periodontal pathogen by Bifurcating gingivalis. Upregulate gene expression of the superperoxide dismutase, which neutralize toxic oxygen metabolites, pH or hydrogen ion concentrations affect their metabolism. In the mouth, pH varies between 6.75 and 7.25. It's maintained by the saliva through saliva flow, power system bicarbonate, phosphate, peptide proteins. In an acid media, in the eco niches are developed lactobacillus, streptococcus mutans. Salivary flow rate and concentration of the buffer component have circadian rhythm, within the slowest flow of the saliva occurring at night. The metabolism of protein leads to the production of amino acid, alkaline pH. Redox potential and anaerobiosis is low oxygen potential subgingival crevice or bucket characterizes the anaerobic environment in the subgingival plaque. This oxidation reduction level is usually expressed as Redox potential, EH. Oxygen is the only one of the main interaction factors which affect the Redox potential. Aerobic as the growth on the surface of the blood agar in the presence of air. Anaerobic as they do not grow on the surface of the blood agar in the presence of air. Facultative are those which grow in aerobic and anaerobic condition. Cabinophilic as the growth of the microorganisms is stimulated by CO2. Macrophilic requires low concentration of oxygen to grow. The Redox potential has been to be decreased from positive 200 MV highly oxidized to 141 highly reduced after 7 days. Early colonizers of dental plaque will utilize the oxygen and produce CO2. The Redox potential of gingival sulcus is normally at 70 MV. In gingivitis FL2, negatively 50 MV. In advanced periodontal disease FL2, negative 300 MV. Metabolism or properties of the particular bacteria will be influenced by the Redox potential of the environment. Periodontal treatment strategy of using Redox agents to raise the Redox potential will make the condition unfavorable for strict anaerobes. The transition of the gram-positive to gram-negative microorganisms observed in the development of dental plaque is mainly governed by certain physiological and environmental changes in the plaque community. As the early colonizers, as we mentioned, as Streptococcus sanguis and Actinomyces viscosus use oxygen and lower the auto tension of the environment, which in favor the growth of anaerobic species. Also, gram-positive species use sugars as an energy source and saliva as a carbon source. Thus, bacteria in mature plaque are anaerobic and asaccharlytic, use amino acids as energy source instead of carbohydrates. What are the nutrients that can be used by bacteria? It will be like as the squamative epithelial cell, gingival fluid, saliva, residual from host food, products of metabolism of other microbial species. Host defense mechanism includes hormonal changes, stress, and genetic factors. The host defense mechanism could be affected by the hormonal change, stresses, and genetic factors. Relationship with the host. Host defense in the mouth. There are many parts in the oral cavity that can prevent the bacterial invasion, causing destruction, like the innate as the epithelial cell, barrier function, innate immunity sensor, toll-like receptors, inflammatory mediators, antimicrobial peptides such as antimicrobial factors which present in saliva, mucosal antibiotics as secretory immunoglobulin A, cell-mediated immunity as a T-cell. In most cases, the host defense tolerate oral bacteria. The predominant relationships are commensal. The host defense mechanism removing the microorganisms through stimulation of the salivary flow, specific protections, non-specific protections as mucin, antimicrobial factors as lysozyme, lactoferrin, salivary peroxidase, histamine-rich peptides, cysteine, leukocytes complement. Major organic components of saliva affect the oral microflora by adsorb to the tooth and form acquired pellicle, act as a primary source of nutrient for the resident microflora, inhibit growth of some exogenous microorganisms, therapy facilitating their clearance. Gingival cervical fluid influences microbial ecology in a number of ways. Gingival clavicular fluid removes non-adherent microbial cells and also introduce components of health defense, especially immunoglobulin and neutrophil. Nutrient fluorescent flora increase gingival clavicular fluid within gingivitis and periodontitis, raise pH to 7.25, 7.25, even such a modest change alter competitive bacteria. Activity of proteases is enhanced in alkaline pH 7.5 to 8. Contain component of health defense. In contrast to saliva, immunoglobulin G is the predominant, also immunoglobulin M and immunoglobulin A complement, leukocytes of which 95% are neutrophil, collagenase and proteases, which drive of phagocytotic host cells and subgingival bacteria. Several of these enzymes are under evaluation as diagnostic marker for periodontal disease. Breakdown of host hemoglobin results in reduction of hemine, which can be used by peripheral gingivalis for metabolism. Increase in steroid hormone, as is considered in pregnancy, can provide nutrient and antagonistic interaction between bacteria may be in the form of competition for nutrients, competition for binding sites, production of substance by one species which limit the growth of another one, as Streptococcus sanguis, which is considered oral health flora, its benefit one, can produce hydrogen peroxide, which inhibits the growth of Aggregatibacter actinomyces comitans, while Aggregatibacter actinomyces comitans produces bacteriocin that inhibits the growth of the Streptococcus sanguis.